Welcome, everybody. My name is Ulf Schlichtmann. I'm a professor at Technical University of Munich in Munich, Germany, and I'm also serving as the CEO of TUMCREATE. TUMCREATE is a research platform of TUM in Singapore at the CREATE campus of National Research Foundation. TUM actually has been active in Singapore for more than 20 years, first with a teaching subsidiary, TUM Asia, which is still going very strong. And then when, oops, that's a bit too quick. When CREATE was started about a dozen years ago, we joined as one of the first 3 university partners of CREATE. In the first 5 years, 2011 to 2016, we tackled the topic of electromobility in tropical megacities, which at that time was very innovative. So we actually built a working taxi from scratch, which you can see here. Then in our second phase, the next 5 years, we focused on how to optimize transport in megacities from a systemic perspective. And every 10 years, the CREATE entities reinvent themselves. So now, since last year, while we're still pursuing some smaller projects in energy and mobility simulation, we focus more on life sciences. Our major program right now is called Proteins for Singapore, where we are working on alternative ways to solve the food crisis which the world is facing and the sustainability crisis. And we have another very exciting project which we want to focus on today, which is called CellFACE, which is a novel way of diagnosing blood. And this is going to be the main event. And for that, I would like to hand over to Qianyu and Kerem, 2 research associates with TUMCREATE, who are working on the CellFACE project and who will now give you an introduction into this project. And then we hope, even though we're a bit late, that we still have time for your questions. So, Kerem and Qianyu, the floor is yours.
Thank you. Okay. Thank you very much, Professor Schlichtmann, for the nice introduction. And also thank you very much for GYSS committee for inviting us. We will today talk about CellFACE project, which is one of the ongoing research project under TUMCREATE. We are focusing on developing a new next generation imaging flow cytometer for revealing some hidden biomarkers which could be potentially used in clinical diagnosis for various diseases. So basically, first, maybe I can talk about who is involved into CellFACE project. So we are a highly interdisciplinary project from various industrial partners and academic contributors. So core team is coming from Technical University of Munich. We have clinicians on board. We have data analysis and AI experts. We have also contributors from NTU for developing microfluidic solutions. Also, we have contributors from AUS and AUH for clinical parts. And for the TUMCREATE research team, right now we are 3 people in this image, but we also have a few more team members till this picture was taken. So my name is Kerem, the person on left. We have Qianyu. She will also introduce you data analysis part, and we have Komal in biomarker analysis part. Basically, the vision and motivation of CellFACE project is actually developing a low-cost, label-free, and quick blood analysis. Because actually, if we look at the recent technology, gold standard technology used in clinical setting, so basically, even though this is quite effective, there are still room to expand. First of all, cost is still a problem. Secondly, label-free analysis are still something that we need to explore because, for example, in the case of hematology analyzers or flow cytometers, labeling procedure is still a very labor-intensive and costly process. And thirdly, since we are not only analyzing signals, which is the case in flow cytometer, for example, but also we can provide cell morphologies, which is actually a unique way because we can directly have a look at cells and then we can detect some abnormalities, which may indicate various sorts of diseases. So at the end, our aim to actually develop some panel, some biomarker panel that can link various cell types, populations to diseases for quick and low-cost diagnostics in clinics. What we are focusing on is actually fever. So fever is actually something we all experience in our lifetimes. But the thing about fever is that it's a very broad, very general symptom. So there are various reasons relying underneath. And this could be simply, for example, when you get a COVID booster, most probably you will have fever because your body responds against a kind of foreign agent. So that's why, for example, you will experience fever. On the other hand, for example, if you have some kind of viral or bacterial infection, still you will have fever. This could be the case in cancer, pneumonia, or maybe some kidney diseases. So again, fever is a very actually basic but also important symptom that we need to address. And actually discriminating the resource or the cause actually has a very, very huge impact in clinical setting. So just to give you some idea, some numbers, actually the 5% of adults and 15% of elderly people, when they apply to emergency, has fever. So fever is actually the second most common complaint among these misdiagnosed or somehow diagnosed, but a very in a long term. Because the investigating fever and finding the exact cause is quite time consuming and costly process. So that's why actually if we find a way to diagnose the underlying reason in a fast, in a low cost manner, actually this could potentially have quite a huge impact in patients' welfare and time. So investigating fever, actually, traditionally, we use blood count, whole blood count. So basically, blood provides us quite a nice overview what's going on in the body because we basically have a look at three distinct type of blood cells, but also we can further analyze, for example, subtype of white blood cells. And in some cases, for example, in leukopenia, which can be caused by viral infection, we see some white blood cell numbers actually increases or simply fluctuates. Or in, for example, the case for leukocytosis, neutrophils can actually fight against bacterial infections so that it also gives us some idea what's going on in the body. But still, actually, these are quite general, broad, maybe indications. And we need something more specific to address what exactly going on in the body. We need some more specific biomarkers. So if we have, for example, look at some blood smear, which is the gold standard for, I guess, for last 150 years, actually, we can also see the variation in number and morphology of cells, which is actually quite a good indicator. But problem with blood smear is that, first of all, this is super labor intensive. It takes time. And secondly, we can only analyze a very small amount of blood. So again, microscopic analysis has been done for quite a long time, but we need something more specific. That's why, for example, flow cytometer has developed, and this actually changed the life science and medical sciences quite a lot. But still, the problem we are facing is these technologies cannot really provide us a low cost and label-free fashion analysis. For analyzing cell morphologies can tell us some abnormalities, in morphology and population size. And also we can link these information into different diseases and different conditions. Oops, sorry. So for further investigating fever, we need to actually differentiate the source of the fever, which could be viral, bacterial, or just simply fungal source. So the problem here is that the discrimination of viral and bacterial infection requires quite a intensive work. That's why actually clinical diagnostic takes some time, which may result delayed diagnosis and delayed treatment. So in the other, for example, there could be other cases like non-infection cases, autoimmune diseases, malignancies, some cancer diseases, some allergy or something like this. So that's why we need to actually differentiate the source. And again, as I mentioned, actually right now the gold standard in clinical setting is using hematology analyzers. So basically hematology analyzers are very good at providing an overview about blood cell count. But the problem here is that they cannot provide, for example, cell aggregates, for example, leukocyte aggregates or platelet aggregates, which might be the case in different types of infections. This is something we need to investigate because these machines can only provide us signals, but not morphology. So as a result, actually, there is a unmet clinical needs that we need to deliver label-free, low-cost, and point-of-care solutions, not only for general healthcare, but especially for emergency department and intensive care. So the processing should be minimum, should be label-free manner, and it should be repeatable. So what we also introduce is actually we can analyze using artificial intelligence so that we can be more precise and quick. And from here, I think Qianyu will take over and then give you a little bit more about technical aspects.
Thank you, Kerem. So now I'd like to talk more from the data analysis part. From this slide, we can see that once we receive the samples from the hospital, we will inject the sample through the digital holographic microscope and do the imaging. Then we're going to measure the blood samples, record the images, and do the analysis. And then from the right side, we can see some examples of the aggregate, like leukocyte aggregate, platelet leukocyte aggregate, or just platelet aggregate. Then in our next slide, we can see from the left side, in this video clip, we get the idea how the DHM microscope record the high throughput blood sample images. And from the right side, in TUM, we have already successfully differentiated 8 different leukocyte subtypes. In the top row, we can see the reconstructed face images of leukocyte subtypes. And the bottom row indicates the corresponding heat maps of the top row face images. And here the scale bars are 5 micrometers. So we can move to the next slide. Here we can see the plot of percent. We can see the we, by applying the deep learning algorithm, we feed the large scale of cell images to the neural network to train the model, and then to perform the object detection, the instance segmentation, and distinct cell types classification. And our target is to find out rare aggregates and quantify them. So with these analyzed statistics, we are able to provide to clinicians to support them for diagnosing the severity of patient diseases. So nextly, we maybe Kerem can take over the slide.
So we would also like to talk about few use cases that we already worked on, which one of which is actually identifying abnormalities in blood, and this might indicate actually leukemia. So for example, for the first figure from A, you see that healthy control samples, this shows quite a 3 different actually population. And when, for example, for acute myeloid leukemia or lymphocytic leukemia, or for example, chronic lymphocytic leukemia, you see actually these populations shift and a little bit more intense. So definitely there is a difference compared to healthy control. So basically just getting white blood cell count from DHM, we can discriminate these diseases. But for example, myeloproliferative neoplasm, the characteristic didn't change much, but still, for example, we can further analyze these data population through our artificial intelligence network to come up with more precise results. So further, maybe Chen, you can continue in this part.
Yeah, thank you. Actually, we can combine this slide together with the next slide. So this is here, this plot, we can see the percentage of platelet aggregate of COVID-19 patients in ICU at the very first measurement day. And it's clearly observed that the mean value of thrombocyte aggregate percentage is lower than the mean value of mid and severe patients aggregate percentage. And actually in the second slide, it also depicts the amount of thrombocyte aggregate percentage at the maximum measurement day in the relationship to total thrombocyte count from the different cohorts. And these 2 plots state that our label-free technology can is greatly helpful for clinical diagnosis to identify whether this patient is infected or not. So yeah, that's all for our this technology from the
yeah. And for example, so far we showed you a few applications, which one of which could be whole blood count, for example, for leukemia in particular. And other is, for example, it's a very recent study that we did in Munich site, actually, the COVID study. So all of which actually showed us that CellFACE can provide us a very unique way to deliver some novel diagnostic information. Because, for example, if you have a look at the charts here, even though fluorescent flow cytometer and hematology analyzers are gold standard in both academia and clinical settings, these technologies actually require some labeling procedures, some the training personnel to use it, and these technologies, for example, cannot provide us cell morphologies that we already showed you. These are unique and important information for diagnosis different diseases. On the other hand, for example, we have regular traditional biomarker analysis, but these actually analysis can fluctuate a lot, especially in very severe infections. And for, as I mentioned, blood smear analysis is very actually good for determining, analyzing cell morphologies, but this is a very label intensive and very kind of limited way to analyze blood samples. So in the intersection of all these, actually what we propose is providing a label-free, low-cost, and hopefully a technology that can provide us a unique and new biomarkers.
So thank you everybody for joining. I hope this has given you some overview of TUMCREATE and CellFACE, and please do feel free to reach out to us if you have further questions. We'll be very happy to answer them. Thank you, everybody.
Yep. Thank you very much.